Okay, so now we're going to move on to the CRISPR Cas9 system, which I'm sure you've heard about because it's one of the more revolutionary techniques of gene editing. In this one, we're mainly going to explain what it is and how it actually works. I'm not sure how long it's going to take, and then if we have time, we're going to talk about some of the genetic diseases that we can actually treat with the CRISPR-Cas9 technology and that we are preparing to treat, but if we don't have time, we'll just do it in the next one. So, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It targets DNA or RNA as a protective measure against invading viruses in concert with Cas9 enzymes. Cas9 is just an RNA-guided endonuclease enzyme. The RNA that guides Cas9 is the CRISPR RNA from the spacers that we're going to talk about in a moment, with part of the palindromic repeats that are homologous to the specific tracer RNA for the system. If that's confusing, don't worry, we're going to go over it now. So, in case you haven't heard the story, the CRISPR-Cas9 system was first discovered by a man called Francisco Mojica, I believe in the 90s, when he was just studying some bacteria that lived in extremely saline conditions. So I think they were just bacteria that lived in really salty hot springs, if I remember correctly. But he noticed that in their genomes they had these weird palindromic repeats that he looked deeper into, and he discovered this system. If you don't know what a palindromic repeat is, it just means it's read the same both sides. So think about like words that are palindromes, bob, level, girafferic. It just means they read the same both sides. So for DNA, a palindrome could be like TAAT if you're talking about 4 base pairs, or maybe AGTA or TATAT. Hopefully that should give the idea. So the first step is adaptation, where the bacterial cell will insert a new viral spacer into the CRISPR locus. So it'll take part of a virus's DNA, it'll cleave it with bacterial enzymes, and it'll chuck that viral DNA inside of the CRISPR locus. And the DNA that it takes from the viruses are going to be the spacers that are shown on the image in the bottom. And these spacers of viral DNA are broken up by the palindromic repeats. Step 2, expression, is when these spacers of viral DNA are transcribed in the CRISPR DNA locus, and then the CRISPR RNA that comes from the transcription of these spacers gets processed. And that's quite a heavy process that we're going to get to on the next slide. And then step 3, interference, detection and degradation of foreign DNA by CRISPR-specific Cas9 proteins. That's where we get this, the CRISPR DNA produced from the spacers. Remember, the spacers are just viral DNA. The spacers get transcribed to produce what we call CRISPR RNA, which is going to be complementary to the viral DNA, so it'll break it down. Or more likely the viral mRNA that's going to be produced, so the cell isn't producing a massive amount of it, kind of like with the RNA interference part. Once the CRISPR RNA is transcribed, It's going to form a complex with Cas9 proteins and tracer RNA that becomes specific to the viral DNA that the spacer came from. So it's how bacterial cells defend themselves against viruses. The CRISPR locus acts as a memory, a genetic memory for these viral infections, kind of like our acquired immune system, storing part of the viral DNA within it as the spacers that help the bacteria fight off future infections from the same virus. So the steps of the CRISPR Cas9 system, this is an in-depth look. If you want to read this in your own time, it might make a bit more sense. But in the bacterial genome, the CRISPR locus acts as the memory of the past viral infections because it's taking the DNA from the viruses and it's chucking it in between these palindromic repeats so it can be transcribed if it comes into contact with the same virus again to produce Cas9 complexes that are specific to that viral DNA. So it's going to destroy the viruses and it'll get rid of the viruses in the bacteria cell. The CRISPR locus spacer is transcribed to make CRISPR RNA, a transactivation RNA, which we call tracer RNA, with homology to the palindromic repeat region is activated. That just kind of means, if we look at this image, we can see the black DNA strand. That would be the thing at the bottom that's kind of curvy on the bottom right is the tracer RNA. I wish I had a laser pen to show it. The part of the black DNA at the bottom that is forming these hydrogen bonds, the purple lines with the blue target sequence, that would be the CRISPR RNA that we just said has been transcribed from the spacer. And then we have this bendy part, the loop of the black strand, where these two, where we see the purple lines representing hydrogen bonds between the two black strands. That is the part where the tracer RNA is homologous to the CRISPR RNA, and it's just this region that's homologous both comes from the palindromic repeat region. So the palindromic repeat region of the tracer RNA pairs with its homologue on the pre-CRISPR RNA, recruiting RNA 3 and Cas9. The individual CRISPR RNAs are separated by RNase 3, resulting in individual complexes of the CRISPR RNA, tracer RNA, and Cas9, which is exactly what we see here. So we've got the Cas9 in a in pink kind of color, and then the black strand, the straight line is the CRISPR RNA, the bendy bit is the tracer RNA. And these are specific to the spacer that the CRISPR RNA was transcribed from. So we now know our viral specific CRISPR RNA made of the complementary spacer and palindromic repeats recognized by the tracer RNA, the endonuclease Cas9 and the recruiting tracer RNA. Each of these complexes seek out the virus which the CRISPR RNA produced by the spacer is complementary to. So the virus that made that spacer, well, the bacteria cleaves part of the virus to make the spacer to protect it from the virus in the future. The CRISPR RNA binds to the protospacer adjacent motif on the target sequence, which is this red part, and it cuts the DNA after this protospacer adjacent motif, which we just call PAM for short. So the Cas9 complex with the tracer RNA and the CRISPR RNA separates the double-stranded target DNA and cleaves it, so it cuts the target DNA after the PAM region, making the target sequence dysfunctional and preventing any viral